Hi, welcome to mitosis lesson number 2, and these are the steps of mitosis. So let's go ahead and begin. Okay, so we're going to talk about the 5 major steps, which are mitosis, are prophase, metaphase, anaphase, telophase, and finally cytokinesis. However, the first stage, known as interphase, really isn't a part of mitosis, but more a critical part of something we'll discuss a little bit later called the cell cycle clock. However, in interphase, it is basically known as a normal cell functions, and during this time, some things are happening. Okay, so first, I want to make a note that at any point you can just pause it, stop the video, go ahead and take the notes you need from this slide. I'm okay with it. This is all taken from the textbook, so it's also there. All right, so we need to talk a little bit about the chromosomes. So chromosomes are really made up of materials we call chromatin. That's these little thin lines here. Chromatin begins to wind up along the DNA, what we call histones. And as they wind up and condense, they're then called chromatids. During interphase, during the synthesis part, the chromatin is replicated and then condenses into 2 identical chromatids. And because these are chromatids, we call these chromosomes. So basically, we interchange the word chromatid and chromosome as the same. It's just because this has been replicated, we have 2 identical chromatids. The connection between these 2 chromatids that are identical are known as the centromere. All right, so during prophase, the chromatin, which has been replicated in interphase, begins to condense, and we begin to see them as chromosomes or chromatids. The centrioles, which had been replicated during the synthesis part of interphase, begin moving apart. The centrioles are part of the animal cells. They are not part of plant cells. Prometaphase is the point where the nucleus begins to disappear and the microtubules begin to attach to the centromeres in a thing we call a kinetochore. A kinetochore is just basically a protein that's going to shorten the microtubules that are going to then draw the chromosomes to opposite ends. Metaphase is where the chromosomes align in the middle and the centromeres align along the metaphase plate. And in doing so, the microtubules or spindle fibers will then begin to shorten as the kinetochore moves them or walks them to the poles. In anaphase, those chromosomes move towards opposite ends and the centromeres split. Then in telophase, that's when the chromatids are then forming a new nucleus and the cell begins to pucker or push inwards in the beginning of what we call cytokinesis. At the end of telophase, the 2 cells will have 2 nuclei. The center part will partition, and that phase when the 2 cells divide into 2 identical daughter cells, then it's called cytokinesis. So you have now this process of one single cell going through the process of replicating, going through interphase, prophase, metaphase, anaphase, and telophase, and then going through the last part called cytokinesis. And at the end, you end up with 2 identical cells. Okay, so that is our little mini lesson of mitosis. I would suggest that you go back, reread each of those slides. You don't necessarily have to hear me speak, and in doing so, you'll be ready for Monday's lesson where you will be using the pop beads or toys to model mitosis. Remember to go ahead and take pictures and either make vines or just a series of videos showing each stage of mitosis. All right, thanks. That concludes this lesson.